All right. So, hi, everybody. This is Oliver Medvedik. I'm speaking from the Canberra Laboratory of Cooper Union, where I'm the director of, and this is a bioengineering lab. And welcome to the second Lifespan.io Journal Club. So, the journal that we're reading this week, or today, that we're covering is Marion et al. From the Blasco lab. It's common telomere changes during in vivo reprogramming and early stages of tumorigenesis. So if you recall last week, we basically went over the Del Monte paper, and that paper was very similar, but they looked at something different. They were doing in vivo reprogramming using the Yamanaka factors, and then they were looking at tissue repair in mice. They were looking at mice that were prematurely aged, and they were looking at the epigenome changes at certain locations, the methylation status specifically. And this paper here, they choose to focus. They also do a similar approach. They induce with Yamanaka factors, and they do it in vivo in mice, but instead of looking at all these different changes, they're looking a little bit more specifically at the effects of on telomeres and protein that is required to maintain telomeres called TRF1. So that's the focus of this paper. There's also another key bit of difference in these two papers, and I'm going to go share my screen momentarily. And one of the main differences, here we go, share screen. Where's the picture? Yeah, that's it. That give me a second here. Okay, is that being shared? Okay, good. So if you can see figure 1A, they got a little mouse there with nomenclature underneath that says I4F. That just stands for induced 4 factors, and those are the Yamanaka factors that are required to reprogram cells and cause them to get into a pluripotent state. And they induce it using this drug called doxycycline, just like they did the previous paper. But the difference is they do it much longer. And if you recall the last paper, they pulsed it. So the big difference between the two approaches is that if you have Yamanaka factors on too long, you get a lot more reprogramming, and cells start becoming tumorigenic. So you definitely don't want that if you're going to extend the lifespan of a mouse. So if you want to extend the lifespan of mouse, you clearly want to pulse it much more briefly. And I believe in the last paper, they did it for like 3 days. And then they paused it. Here they do it several times longer, 2.5 weeks. So these mice, just keep that in mind when we go over the data. These mice have these factors expressed at a much higher and longer level than in the previous paper. So just keeping that in mind. All right, so let's look at the images here. So you'll notice a lot of colorful pictures here. There's OCT4, telomere fish, merge, DAPI, and I just want to mention that you're going to be seeing a lot of this in this paper, and this is basically labeling with antibodies, right? So this OCT4, there's an antibody against OCT4, and it's causing it to fluoresce green. These are tissue slices, so these are stain sections, so this is immunohistochemistry. So basically what you're doing is you're taking tissues from the organism that's been sacrificed, in this case the mouse, and here we have a large intestine, down here we have pancreas, and you're looking at different regions, and then you're staining with different stains. DAPI is staining for nuclear DNA, OCT4 obviously for the protein OCT4, which is indicative of reprogramming, and then they have something called telomere fish, which is just fluorescence in situ hybridization. And that's basically, they're using a oligo that's been labeled with a fluorescent compound that will stick to telomeres, right? So if you have telomeres, the longer the telomeres, the brighter the dot. The more telomeres, the more speckles you see here. And this dotted line basically indicates the region that they quantified, and particularly the region that is expressing OCT4, and hence the region that has been reprogrammed. So they're using OCT4 expression as a proxy for reprogramming here. And then merge just merges all the regions. Okay. So is there any questions anybody has about this kind of approach. All right. Everybody can see my face, right? All right, good. I just give a little FaceTime here, come back to the paper. Let's see here, share screen. Let's go back to the paper. All right. So before we go any further, I'm going to zoom to the end because they have a model that they've drawn up of kind of what the results are, and then we'll kind of backtrack and look at the results and see how this plays out. So this is a model for the role of in vivo reprogramming in tissue repair and cancer initiation. So the gist of this paper, just to kind of focus and give a summary of the abstract. Differentiated cells have a lot of things in common, and here's a couple that they looked at, very few things they looked at, which is they have short telomeres, and they have no telomerase expression, and they have very low levels of this protein TRF1. If you differentiate, if you do in vivo reprogramming via the Yamanaka factors, or you already have adult stem cells inside the body, and here they can go back and they differentiate and get to this stage, or cells will transform and become massively dedifferentiated and become tumors, you end up with having longer telomeres. Obviously, telomerase expression, as long as it's telomerase dependent. We might do a paper later on another pathway that's used to extend telomeres called ALT, or alternative lengthening of telomeres. That happens in a minority, about 10% of cancers, but most rely on telomerase. And you have high TRF1 levels. So before we go further, what is this protein TRF1 that they look at very carefully, kind of in various locations? So TRF1 turns out to be a protein that is bound to telomeres. And it's actually, if you look at a lot of other papers, it's correlated with shorter telomeres rather than longer telomeres. So if you overexpress TRF1, you get shorter telomeres. And it sort of competes with the catalytic subunits of telomerase. So it's interesting that you have this upregulation of TRF1 that is correlating with longer telomeres in this paper, as we'll see. So that raises a big question to me. What's happening with TRF1 in these cells that are being dedifferentiated? You know, obviously there's big differences here than what's going on in paper as far as how the cells are treated, but it'd be interesting to me. That raises a big question, like what's going on with TRF1. Maybe there's a paper out there that follows up on that, but if somebody has the answer, I am all ears. Okay, so let's look at the first experiment that they've done. Figure 1, telomeres elongate during in vivo reprogramming. All right, so that's the conclusion from this data. And what data do we see? So figure B, basically, I went over kind of the background of the staining. Telomere fish. So they look at two different regions, two different organs of the mouse, large intestine and down below, pancreas. And the dotted region is the region that correlates to reprogramming. And notice that I believe in this paper, just like the previous paper, these Yamanaka factors were induced throughout the cell. There's a doxycycline inducible promoter that's in all of these cells, but you only have certain regions where you get induction. And again, maybe there's a simple answer to this. If somebody knows the answer, why certain regions, why not all regions, have a low level of, if they're all been induced with doxycycline, why is there these regions that are not, I guess, upregulating OCT4? That's another question. I'd be interested, are they senescent cells? Are they, something else? Or maybe there's just a trivial answer to that. So those just right off the bat, I have those two questions I'm going to pose to the audience. So what they basically do in both of these figures is, what you do is you put this under a fluorescence microscope, you start taking images, and then you use image processing software to basically count the speckles, or look at the intensity of the speckles. That's basically what you do. And then you get a bar graph. So what they're looking at here is mean telomere intensity. So basically, how bright are these red dots? And these are arbitrary units, spots. And they look at epithelial non-reprogrammed, which is this region that's not, boundary. There's no little dotted boundary around. And then there's the reprogrammed region, which is OCT4 positive, which is this region here. So they look at N equals 1,434. So these are how many nuclei they looked at, and the nuclei correlate to these blue dots here. So they say the p-value is 0.001. So when they do this counting, and it's really hard to see here. I mean, that's if I have one main complaint, it's for me, at least visually, it's hard for me to correlate here with these bar graphs. That's just me. So according to their raw data, this is what you get. You have slightly more, I guess, mean telomere intensity in reprogrammed OCT4 positive cells. I don't know so this is intensity, remember, this is brightness. So it's hard to find out really from this figure how long the telomeres are. What does the intensity correlate to? Is that, an extra kb? Is that extra 5 kbs? But it is brighter, right? So that's the correlation that's made there. And you get a similar effect when you look at pancreas, particularly when you looked in this OCT4 region versus this region that's non-stained. So, yeah. So, little issues. When you do this sort of immunofluorescence, it's sometimes tricky to delineate the regions. I'm just going to nitpick a little bit. So these merged images with, DAPI here and DAPI here, appears, it looks a little less, I don't know, at least on my image, looks a little less focused here, a little sharper here. I mean, this is the same image, right? So it's harder for me to kind of overlap these cells when I'm looking at them. But again, once again, these bar graphs correlate to the data that you see there. So they do elongate to an extent. How long, I'm not sure. Then they look at TERK expression, and TERK is, forgot what it stands for, but it's one of the subunits of telomerase that's required for telomerase to be catalytically active, and I believe it's the RNA subunit. And again, just to refresh everybody, telomerase is an enzyme that's required to replicate the ends of chromosomes. It's basically part of the reverse transcriptase family. It uses, it's a ribonucleoprotein that carries around its own template in the form of an RNA. So there seem to be a lot of really interesting ribonucleoproteins out there. CRISPRs, dicers, ribosomes. Maybe we'll do an entire, I don't know, review of ribonucleoproteins at one point. Anyway, so here they're showing that TERC expression increases during in vivo reprogramming. So we saw that slight increase in telomere length up there, or intensity of foci. Here what they're showing is, again, basically immunofluorescence staining. You have cells that are pancreas I4F, and those are the regular, that's the ones we looked at up above. And then they have these other cells from another mouse strain that they crossed. And these are the Yamanaka factor induced mouse system, but in a mouse background that's not expressing. So it's a homozygous double negative telomerase, so it's missing TERK. So there's no telomerase here. They can't extend their telomeres unless they do ALT or some other pathway, which is fairly rare. So this is sort of a negative control here. So again, you have TERK expression levels go up. So OCT4 induction. So they're looking at this boxed region. This boxed region, again, is the region that, and here's a sub-region here, and here's the region that's outlined. And the OCT4 expression corresponds to, again, by proxy to the reprogrammed cells. DAPI again for nuclei, and then TERC, TERK for telomerase expression. I don't know why you have these little background spots here. I mean, these are TERK minus cells. They're looking at this. So I'm, that could be indicative. So you see a lot of less TERC here. I mean, that's, you get background with this type of staining, but again, I would think if it's TERK double minus, you get substantially less. But it could be just a lot of background hybridization taking place. So if we go back here, they look at this region here, Turk. I'm not sure what these little white dots are. I looked through the paper. I couldn't figure out what these correspond to. So it could be a typo. Does anybody remember what these little white dots were? I can't. It's not in the figure legend. I'm just gonna ignore them.
They're little white dots.
They're little white dots. But initially I thought they corresponded to like indicators of telomere sites, but they're not. So I don't know what that is. So here's the boxed in region that they look at here. So this is the zoom in and this is the zoom in, right? And over here is OCT4 expression. So what you're basically seeing is that you get reprogramming, and evidently you get more TRK spots. So again, here's nuclei. They look at 344 nuclei, and it's hard for me to count those spots and those figures up there. That's unfortunate for me, but you get 0.4 spots per nuclei. So it's, again, they're not getting that many, that much telomerase expression, but it's higher than the region that is not OCT4, right? So this corresponds to the OCT4 region, the reprogram region versus the non-reprogram region, and this is their control. So if they look at the OCT4 region versus the non-OCT4 region, even despite this background, so the background cancels out, which even though it's a high background, and they say that this is the low kind of basal number. So you don't have so again, you get OCT4 expression. It's not dependent on telomerase there, but TRK expression increases during in vivo reprogramming. Any questions at this point? Let me kind of pause here. Oops. Let me stop sharing.
I'm just having a look now.
Yeah.
I'm checking a couple of channels.
Mm-hmm.
Nothing yet. That's either a good thing or a bad thing.
Yeah. Okay.
I'm going to assume good.
All right. So let's go back to the paper. Let me zoom out of my face here. And there we go. All right. So C basically looks at now expression levels of TRK and TRF1 and OCT4. The black bar is non-reprogrammed region. The gray is programmed. I'm not sure why the N values here are way less, because you got nuclei 1049, you have 344, and here you have N equals 3, N equals 3, N equals 2. I'm assuming that this is expression levels per cell, as measured by qRT-PCR. Okay, so it could just be different regions of those regions, and these are qRT-PCR samples. So we have N equals 3, N equals 2, and these are the p-values, less than 0.05. So these values are statistically significant. This is sort of on the threshold. Again, these values are arbitrary, right? So this just means that you have a confidence of 95%. It means if you repeat these experiments 100 times, 5 times, it probably won't give you the answer you thought you got. But this is usually, statistically considered sort of the cutoff point for many people. So you get higher expression levels. Okay. So moving on to figure 3, they show that telomere elongation during in vivo reprogramming depends on telomerase activity. That's not too surprising to me. I would be surprised if it was otherwise, because that would indicate that there's a massive upregulation of the ALT pathway. But again, similar images here. You have these OCT4 regions that have been successfully reprogrammed, kind of, surrounded by, I'd be interested to see how, again, like, I can't help but ask myself, like, why are these clusters like this isolated? And if I zoomed out, like, how would it look like, right? Like these clusters that are reprogrammed, are they Are these cells particularly easy to reprogram inside the cell? Are those cells inhibited from reprogramming? Are these like, you know, I'm just rampant speculation here. Are these niches for, you know, stem cell niches? Niches? No, Nietzsche was a philosopher. I don't know. So anyway, so this is what they use for their correlation. Again, the boxed-in region corresponds to the OCT4 expression. The DAPI are nuclei, and then there's a merging. And again, they're looking at, these are the Turk, remember, these are the Turk double minus cells. So, and these are telomere fish. So they're looking at telomeres in cells that do not express telomerase, or at least that catalytic subunit. The same thing for pancreas. So they look at non-reprogrammed and reprogrammed. I don't think there's a statistical significance here. Mean telomere intensity. Same over here. Mean telomere intensity, non-reprogrammed, reprogrammed. And percentage increase in telomere length. Again, this is previous data, and you could see that the telomere length is, you know, percentage-wise, is much less, as you would expect. And I'm assuming that this percentage increase in telomere length is, again, derived from the intensity of the fluorescence image rather than actual measurement via a gel, or intensity probably through a QRT-PCR system. So, so that's the result. Again, not too shocking for me. So this image here is, or this figure here is probably the last one before they start moving on to what happens in cells that are cancerous. So remember, they are heavily dosing these cells with doxycycline, so they're really expressing these Yamanaka factors. I'm trying to look for the figure legend for this guy down here. Where's the figure legend? Ah, here it is. Figure 4. Increased TRF1 expression during in vivo reprogramming correlates with OCT4 expression and is uncoupled from telomere length. So that's probably really the kind of the most interesting part to me, that even though TRF1 is required to maintain telomeres, particularly negatively regulate them, in this case, it is the increased expression is uncoupled from telomere length. So you could have different telomere sizes, and it doesn't matter on TRF1 expression, and it correlates with OCT4 expression. So if you have, wherever you have TRF expression, you have OCT4 expression, right? So it's correlates with the induction of the Yamanaka factors. So here we look at, again, fluorescence in situ hybridization. We have OCT4, and these are the nice bright regions that are reprogrammed. Here it's surrounded with TRF1. You could see here, it's actually, to me now, it's really clear. I could see, you know, much brighter TRF1 in these regions. So, and then DAPI and then merge. So that to me is going to be clear that you're going to have correlation of TRF1 expression with regions that are de-differentiated. And this is the pancreas, right? So, and this is basically looking at, this is just the data made numerical. So TRF1 intensity spots per nucleus, epithelial non-reprogrammed, less, higher in the nucleus with OCT4, much tighter p-value, and you can even see that by eye. So that's pretty obvious. Now we look at the cells that are, the mice that have been crossed with a Turk double minus, pancreas and stomach. Again, box the region in that's OCT4 positive. I just keep asking myself, what's really going on? Why is that differentiated and that's not differentiated? Might be a simple answer, might not be a simple answer. But anyway, we'll later look in the discussion if that was kind of broached, because I don't recall. But nevertheless, again, you have high TRF levels, and you have high TRF levels, and you don't have any telomerase. expressed, right? So TRF levels are upstream of what's happening at the telomeres, clearly. So the Yamanaka factors somehow regulate the expression directly or indirectly of TRF1. That's what it seems like at this point here. So I'm gonna stop sharing and I don't know, pause here for a little moment for everybody to get their bearings. Take any questions, if there are any.
There's a frenzy of typing on Discord, but no one's actually said anything yet. So someone's writing a novel. But what I don't really understand here is if TRF is usually indicative of telomere attrition,
Yeah.
how come in this case it suggests the opposite? Paradox? Oh, hang on. Oh, we have a question. As well as that question, in the stains, is intensity correlated to telomere length?
I would say yes, because it's a hybridization. So there, you would have more oligos that are conjugated to the fluorophore hybridized. So if more of them hybridize, you know, I would say that correlates with longer telomeres. Unless there's another reason why they hybridize with higher intensity.
What's going on with TRF? Is it context specific, dependent on other interactions?
Yeah, I don't know what else it interacts with, right? Because you are expressing these factors.
Oh.
You are.
My wife was disappointed there by the sound of it.
Yeah, she's also confused by the TRF1 result. Yeah, I don't know. I mean, you've got de-differentiation taking place here, so you know, I don't want to speculate. I mean, maybe it's complexed with something. Maybe TRF1 is being used to regulate other genes. I don't know. That's just, I'm talking out of my, you know, I don't. That's hand waving. But there's no, you know, we can take a look in the discussion and see what is suggested there, but I'm not entirely certain. Still unknown to me. I am intrigued by these pockets of de-differentiation and differentiation that are in close proximity to one another. Again, it could be a simple answer to that, and I'm just totally confused, but it just obviously caught my eye. All right, I'm going to go back to the paper now. Let's see, stop sharing my screen.
We might have another question brewing for you at the moment about p-values. But it's a very long thing, and I've just said, well, is it a question? Because they're not sure whether it's a question yet. So them brew. We'll let them brew on that for a moment. I'm not going to go over, you know, standard error, and you know, because I'm a bit rusty on that. I'm not. I'm sure there's a Khan Academy video on how to calculate p-values. It's not that difficult. You just have to have a statistically relevant sample, and I think physicists in general want to see p-values that are like 0.00000 to, you know, 10 to the negative 10. And, you know, when experiments get a bit messier, people are satisfied with p-values that are closer to 0.05. But I kind of half jokingly said to people that, well, that really literally means that if everybody did experiments in biomedical research, like with 100% accuracy, right? And they all had p-values of 0.05. That means that like 5% of all medical journals have the completely opposite result or negative result that they expect. All right. And the question is, there is a question, why are biologists still obsessed with p-values? Can we discuss this a bit? They are abused and generally bad for science.
Yeah.
Apparently. That was someone called The Brain from our Discord community.
Well, I think the problem in general is that, and again, there's been, you know, statistics books have talked about this, that there's just not that many
people actually doing, applying good statistics to their research. And I put myself in that camp at times. And, perhaps, to kind of make it all very much more rigorous, there should be at least one statistician for each paper. I'm not saying that these results here are statistically irrelevant. I'm just saying that that's something that that could be abused, right? But you hope it's not. And that's, without me kind of going on a longer discourse, I think I'm going to just cap it at that. And of course, you have to pick the right statistical tools for the right model, right? P-values just happen to be very easy to calculate based on the sample size. Okay, but not always the appropriate tool to use. But in this case, I believe it is. All right, so I'm going to stop sharing my screen here. Oops, just kind of got lost here for a moment. Share screen. Where's the stop share? I'm looking for my stop share. I can't see my stop share anywhere. Technical problem. Could be. If it's minimized, I have no idea where this darn thing is. I'm clicking on the boxes. There's no stop sharing. I'm clicking on my face. Just mute my audio. Share. Click on share screen. And I can only share my screen. Oh, there we go. I guess you don't have to stop that. All right. I didn't get that. All right, let's go back. There it is. Popped up. Yeah, got it. Yeah. All right. So in these last two figures, what they're looking at is they're looking at regions that are, I guess these are different regions. I don't know if these regions correlate to those regions that we saw up there very well. So remember, I kept harping about these boxed-in regions that have a lot of dedifferentiated cells because of OCT4, and here they're looking at a different kind of morphology or phenotype, and this is dysplasia or dysplastic area, which is a term used in basically means, I guess, an area that is poorly differentiated, indicative of cancer. And they want to basically look at, well, I'll just read the title, in vivo treatment with TRF1 inhibitor. So here they have an drug that will block TRF1, ETP47037, reduces dysplasia. The thought here being that TRF1, if it's required for this reprogramming, they look at dysplasia. By inhibiting it, it actually reduces dysplasia during in vivo reprogramming without affecting telomere length. So here's another kind of conundrum to me, is that you're blocking TRF1 with this inhibitor. There's no effect on telomere length, and it's reducing dysplasia. So it's downstream, perhaps, of the Yamanaka factors, but TRF1 seems to be playing a role in reprogramming, at least, directly, at least in these cancer cells. Well, maybe indirectly, but at least in these cells that are dysplastic. And I have to admit, I'm not a pathohistologist. I know this is a H&E stain called hematoxylin eosin. It's basically, a very classical stain that is used to stain acidic chemicals like DNA and basic molecules such as certain, amino acids like arginine. So you get, this counter stain where you have basic and acidic regions are either dark purple or pink, right? So this is just a non-specific stain. It just shows you overall morphology of the cells. So these are cross sections, and these arrows are pointing to dysplastic areas. And I'm not a histopathologist, so I couldn't tell you what a dysplastic cell looked like versus a non-dysplastic cell. I'm gonna have to go with whoever was more experienced and indicating that these are dysplastic cells. So here they do have a cross section of these cells that are have the Yamanaka factors expressed. Also, they have labeled TRF1, I believe.
have to go back there and double check why. And here is the same, well, not the same slides, but the same cross sections from the similar regions, but have been blocked with ETP47037. And they say that these areas, I mean, this clearly looks different to me, but maybe the dysplastic area is less dense, but you have less dysplasia according to histopathology here. And again, this is the numbers that they have here. Large intestine, again, p-value 0.05, n equals 3, n equals 3. Vehicle, ETP4707, so percent goes down. Non-dysplastic cells. So these are cells that they look at mean telomere intensity now of cells that are non-dysplastic, and there's no difference, right, in telomere intensity, which is, again, a bit odd considering that, you know, TRF1 is known to have this effect when you overexpress it. And I'm forgetting what the EGFP TRF1 plus K was. I'm gonna double remind myself, just blanking on that. Clearly a GFP labeled TRF1. They do have a system where they have an consistent, perpetually active, you know, RAS protein, but I'm not sure if that's the same one. I'll go back to that a little later. But anyway, they say this is non-significant here. They say percentage increase in telomere length, dysplastic versus normal cells. N equals 2, okay. Versus sample sizes, you know, again, I'd have to check with a statistician, but take that, you know, for what it's worth. This is non-significant. This is non-significant either. The mean telomere intensity. So if you're blocking TRF1, you're not really having any effect on telomere intensity, which is interesting in these cells. I'm not sure what to make of that. And finally, oh, here's the constitutively active RAS. So these are, so they're looking at a very specific type of cancer called acinar ductal metaplasias. These are lesions found in the pancreas, and they also see increased TRF1 expression. And these are these ELAS KRAS G12V mice, which is just basically a point mutation that makes RAS, which is a proto-oncogene, oncogenic, and it basically just sort of makes the signaling system downstream just fire continuously, and it causes cancers. So again, H&E staining from this region. CK19 is indicative of this change. This is cytokeratin 19, a protein that accumulates in cells that undergo this acinar to ductal transition. So these are the cancerous cells when you have elevated ELAS KRAS G12. Again, it'd be interesting to see why these other regions aren't being turned on. But similar to the other figures, here are regions that they look at. So these regions that they circle are the regions that have undergone that transition. This orange boxed in region hasn't. This is TRF1 expression. This is DAPI expression. This is merging. So what they're going to do is they're going to correlate TRF1 levels with DAPI, right? And see basically, and see the difference between these regions versus these regions. And what's this down here? TRF1 CK9. This is just a different region. I think this is just a different region. Okay. Oh, actually, no. This is like a variant of this cancer where this kind of lesioning happens. That's called pannins. I'm not gonna. If they have this acronym spelled out here. I'll read it, but they don't. So it's basically lesions that happen as a result of tumorigenesis. So they look at both regions and they quantify TRF intensity, spots per nucleus, and again, they say they get a higher number here for metaplasias when they count, you know, n equals 8 versus normal acini, which are the cells that are not, you know, transformed. And looking at TRF1 intensity spots per nucleus, this is interesting here. I don't know, they say in some regions you have increased TRF1, in other regions you don't, and they mentioned that in the paper, that like 65% of the regions that are these lesion regions have TRF1 up and other regions don't. Again, I don't know why. That would be a great follow-up experiment. So overall, overall you are getting TRF1 levels correlate with more dysplasia, according to this data here. And you are getting TRF level 1 levels go up with cells that are dedifferentiated when you look via OCT4. And evidently TRF1 during this dedifferentiation process, as opposed to the normal process of cells, you know, just hanging around, it's not having a direct effect on telomeres. Even though telomere length does go up when cells are becoming dedifferentiated. So this paper here, you know, poses a lot of intriguing questions at the end. What's TRF1 doing? Why is it so important? And I don't know the answers to that. So let's scroll down here. Just kind of looking through the discussion. All right, so that was the paper. I did not go over, please note that there are supplementary data online that the paper referenced, looking at different regions of, you know, different cellular regions. So if you want to dive into that, you feel free to go ahead. This is an open journal, so you shouldn't have any problems finding this paper online. And are there any questions? So what's TRF1 doing?
It's TRF doing, as I said earlier, you know, it seems to be a paradox.
Yeah, I mean, paradoxes are good. It just means that something cool is going to pop up at some point. You know, nothing is, and again, like a lot of the earlier stuff, you know, you try to extrapolate as much info as you can, but you know, you are talking about overexpressing TRF1. So if you overexpress it, telomeres get shrunk. So is there some other negative feedback loop that's happening and that's not dependent on TRF1? You know, that's the overexpressed, is that signal something else to shrink the telomeres? I'm not 100% certain. But from the published literature that I can gather, TRF1 binds directly to within the complex of proteins that bind to telomerase. It's a small protein. It's a couple of alpha helices, and it just decorates the ends of telomeres, and it has basically blocks the activity of telomerase, the catalytic telomerase. So that's what I've picked up from other journals.
Well, like everything in biology, it'll be case specific, won't it? There's a lot of stuff. You know, there's a lot of stuff that's beneficial in small amounts, but detrimental, and vice versa. I mean, look at
look at these Yamanaka factors, right? Yeah.
I mean, there's loads of stuff. Things like TGF beta is a classic example. At sort of normal physiological levels, it's okay, but once it reaches that tipping point, it becomes a problem. You know.
Yeah.
So biology, don't you just love the spaghetti?
Well, we could just say that everything has to walk the narrow path, right? There's this little buffer zone that we all dwell in that's homeostasis maintains, and veer off this way or that way, and bad things happen. Bad thing meaning equilibrium. We don't want that.
It's true, isn't it, really? I mean, too much of a good thing, too much of a bad thing. We've got a question here. Wouldn't making chromosomes regrow telomeres, brackets generate telomeres, increase the chance of cancer the way cells that don't die do? How can we make sure not some chromosomes get telomere regrowth therapy?
Yeah, I think that's a good question. I mean, there's certainly, there's papers out there that suggest that, it's not a problem. There's papers suggest that it is a problem. Certainly you need, if you're going to have a cell that's going to be dividing continuously and has linear chromosomes, as all of our cells do, then, it could be a problem if you have telomerase being constitutively upregulated. And that definitely is a worry. I certainly wouldn't be just randomly upregulating my telomerase levels, in my cells in the hopes of, becoming perpetually youthful. I think there's a lot more going on than just that. And as far as upregulating telomerase willy-nilly, I think that's a good point. We are seeing telomerase, in these papers kind of shut down or turned on all over the place, but it would be interesting. I don't know of any papers that actually specifically target telomerase transgenically in certain cell types, like somatic stem cells, because I have to take a look. It probably might be a ton of stuff out there on that, but I'm tending to agree with you on that, in that, you don't want telomerase on in terminally differentiated cells, nor do you need it on. So yeah, if you can focus it just on somatic stem cells, assuming that there's not other things going wrong with somatic stem cells as they age, which probably is the case, rather than just telomere shortening. Yeah, it would make sense to me that you would want targeted expression rather than a global expression upregulation in every cell, including cells that are quiescent, which would be pointless unless telomerase has all sorts of other functions, which it might.
I don't know, there's evidence to suggest that stem cells don't age quite the same way as the rest of cells. The work by Irina Conboy and Michael Conboy certainly strongly suggests that when they rejuvenate stem cell populations, functionally, there's very little difference. I had a conversation with her about 2 years ago.
What do you mean by very little difference between what?
Well, when basically what she was saying was when they rejuvenated the this was actually in relation to a TGF beta paper from 2015. She was saying that once the signaling environment is corrected, she uses the word calibrated, they found that stem cells became, functionally almost the same as young stem cells. And a lot of the markers, the aging markers were gone. So she seems to be of the mind that there's not a lot of difference between an old and a young stem cell. And given the correct sort of environment, that there's really not much difference between them.
Well, what do you mean by no difference? You said, but how can you even tell a young and an old if they're undifferentiated? Like she's saying that there are no old stem cells, or what?
Well,
It's the
signaling environment. Signaling. I can't say that word. The signaling environment tends to sort of dictate. Hang on, let me kill someone. It tends to dictate whether how they function to a greater extent. But yeah, the rejuvenated stem cells they found, there was very little difference between, old and a young.
How could they tell that they're rejuvenated? Whatever they're measuring for rejuvenation, if they couldn't tell between old and young?
Well, I mean, they looked at a variety of markers and things like that, but they, I'd have to get the TGF paper But they looked at obviously gene expression profiles and
Something's changing, right?
Yeah, I mean they're not exactly perfect,
But
you know,
is she saying the telomeres don't re-extend? Is that one of the things?
They didn't look at that actually,
Yeah.
but she was of the mind that that they probably did, because I suggested to her at the time, and this is a couple of years ago. I said, do you think that telomeres reset, you know, when you stabilize the signaling environment? She believed that they do, and that would probably make sense, you know.
Well, certainly this paper suggests it.
Yeah, I mean, you know, if you stabilize the what I call the signaling environment, I think Hallmarks calls it altered signaling environment. I can't remember what they call it, but it would make sense because, I mean, after all the hallmarks are sort of linked together, aren't they? So signaling environment is going to be influencing epigenetic profile, and if you improve the signaling environment, ergo, you may well improve the you know, the epigenetic profile of the cell, and in which case it becomes functionally ill. And we know this from the parabiosis work that Irina did as well, that this happens to a certain extent. Now, which comes first, the chicken or the egg? Now, that's another question. But there's no doubt about the two being linked, but, you know, that's not news, really. We know all the hallmarks are linked. You pull one and it pulls the others, you know? So,
well, I'm overall, I'm pretty impressed that you can using just pulsing of these, you know, cellular reprogramming with Yamanaka factors, and you can have a positive effect on the mice. That basically the epigenome very strongly suggestive that it's not just correlating, but it's also a causation there, right? That so I find that to be really interesting. The other thing that sort of wasn't quite addressed in the last paper that I'm kind of nitpicking, I mentioned this to Keith earlier, is that when you do this reprogramming, you have a population of cells, and you have cells that reprogram, and you know, and they reset, and you call them rejuvenated because you look at factors like telomeres, beta galactosidase expression, and all this other stuff, and they look and act new. But is this happening for a single cell, right? Or is this a population effect? Is it just that, you know, when the cells divide, you know, the crappy ones die off, and then you're left with a, you know, selected sample of cells that are rejuvenated? Or can you have a quiescent cell, and you do this, and the cell, that cell, without undergoing division, rejuvenates? I'm just kind of tossing that out there because, you know, there's also work that strongly suggests that aging could be a problem of, well, at least one aspect, right? And I think it's covered by, Aubrey went over it, is asymmetric segregation of damaged factors, right? When cells divide, they push garbage into one direction. So it'd be interesting to see it from a single cell perspective. I mean, the tools and technology has gotten there where we can do that and see what's happening if you, you know, for that one cell, right? Is division required? I guess what I'm getting at is division required for, or something similar, or autophagy or whatever, for this rejuvenation to take place, right? Like, what does it mean? How does it rejuvenate, right? Is it done within a cell, and then when both cells divide, they're both rejuvenated? Or is something else occurring? I guess I'm trying to drill down here kind of deep. Anyway. All right. Well, are there any other questions out there floating in space?
Just having a look now. Obviously, I'm monitoring multiple channels. I don't see anything. Oh, hang on. Oh, I don't see anything, he says. And the second I say it, one pops up.
Okay. So maybe we'll take one more question and kind of wrap up. It's been going for a nice solid hour.
Yeah, it says, the question is, it's newborn's umbilical cord seems to have a lot of stem cells. But how can we get enough cells in a group? Fat? Question mark?
How can we get enough cells in the what?
In a group.
In a group? I'm not following.
No, let's just check. Let's just get clarification.
I think well, if I'm not mistaken, I think he means how do we get, is that enough stem cells? And the answer would be yes. If they're if they're basically in a undifferentiated state and they're expressing telomerase, you should propagate quite a large amount of them. And then, of course, the technology now developing with reprogramming and Yamanaka factors, you shouldn't be in any worries of depleting that population. I can't hear you. I can't.
Can you hear me now?
Yep.
Yeah, he's saying he didn't mean a group. He meant an adult. In other words, there's lots in umbilical cord, for example, but how can we harvest enough stem cells from an adult? And he's saying, is adipose tissue a possibility, or that seems to be quite popular?
Well, yeah. I mean, it's certainly very easy to get from liposuction clinics, relatively speaking, than brain tissue. So, that's probably the major reason why people go for adipose. And but certainly you would need to do some extra treatments, right? Like you would have to go through this. I would think you'd have to go through some rounds of this reprogramming if you wanted to get enough of them, right? I don't know what the ratio is right now when people isolate adipose-derived stem cells versus cells that don't have any stem-like characteristics. So that's something I'm going to look up, what the ratio is, So, I guess there's two ways to do it, right? One way is to basically harvest all the cells and then de-differentiate them using Yamanaka factors. The other approach is just get really, really, really fat and just isolate.
No.
And then you have a large sample pool.
Don't try that at home, folks. It's not really a very good longevity strategy. There's good evidence to show that adipose tissue in particular aggravates senescent cells. We can talk about that next time, actually. I've got a couple of papers about that. How fat literally is killing you on a senescent cell basis. But adipose tissue is quite a common tissue for harvesting MSCs, for example. I know a lot of people, mesenchymal stem cells, and they often take them from fat. So I mean, it makes sense, really. You get the benefit of removing the fat, and you get some use out of it.
Yeah, but just going back to being on the balance point here, right? You still need adipose tissue. It's there to cushion your organs and provide fuel. So don't get all anorexic.
Yeah, well, I mean, again, it's back to homeostasis, isn't it?
Yeah.
I mean, it's that balance. And we have one last question, if we will.
All right.
He says, do you think stem cells can be used to replace damaged brain cells in cerebral palsy patients to restore motor function, for example?
Well, I'm going to say yes, and I don't know if I don't know specifically for that, but I do know that stem cells have been used, I think, successfully in some experimental treatments for certain degenerative diseases of the brain. I'm not sure. I think might be Parkinson's. I think maybe there was they had them redifferentiate to dopamine producing cells.
That's right.
Yeah.
I think I mentioned that during the panel we had with Aubrey and Alex. It was where they, in mice, but hopefully move into people. But they, what they did was it's very similar to what we've been talking about today. Inside a living animal, they literally used factors to reprogram. I think it was astrocytes, which are fairly common in the brain. And what they did was they sent astrocytes a series of instructions delivered by, I think it was a viral vector, and told them basically to change the type of cell they are. And within 5 weeks, and I'll send you the paper, they had a massive, a very significant amount of dopamine dopamine producing neurons had appeared. So the population, went up really quickly as the astrocytes changed from one cell type to another. So it's looking increasingly to be the case of, rather than what they've been trying since the 1980s to actually, do things ex vivo and put them back into the brain.
Right.
They seem to be wanting to reprogram in situ, and it seems to be a very good approach.
Yeah. And I'm going to just kind of mention a disclaimer out there for stem cell therapies, and I've read some papers about this. Not every stem cell is the same, right? You have different somatic stem cells, and they both have, so there's been some controversy, for example, at some clinics are operating under the radar in Florida, and they've blinded people by injecting fat-derived stem cells into their retinas in the hopes that they would differentiate into retinas, which did not happen. You just had just random growth and hemorrhaging back there. You have to really coax cells to, you can't just put a cell in one environment and hope it turns into an astrocyte or into a neuron, right? It has to be tweaked specifically, and some somatic stem cells will not differentiate into other types of tissue, right? You have a limited kind of range. So, and there is a paper out there where they did successfully, one patient, so sample size 1, an elderly Japanese woman, get retinal stem cells, or I don't know what they used to derive them, but the proper ones, and it successfully engrafted. But she still didn't get back her vision, but there was no other effects. So, it's kind of a neutral response. So it's doable, but they don't, nobody's, if you have a retinal degeneration, I don't think that's been, that particular blindness has not been tackled with stem cells. So be wary that there is a lot of clinics out there, stem cell clinic, that basically are using you as a guinea pig and asking for tons of money. Not all cells are alike. The devil is in the details.
And not all clinics are alike. So, if anybody's thinking about getting, stem cell therapy, be very careful. It is relatively new. And caution is always to be advised. We're all keen on getting this done, and longevity, but there's nothing more embarrassing, Oliver, than actually accidentally killing yourself through a misguided experiment.
Right, or making yourself blind permanently. Yeah,
exactly. Just before the actual technologies pass through safe clinical trials and arrive. I mean, can you imagine how embarrassing? You'd never hear the last of it if you killed yourself 6 months before SENS.
Yeah, I'd be regretting it for a long time.
I'd be like, I can't believe that guy. It's like 2 months later, the technology arrives. Why didn't they wait? But we're getting really close. But, there are a lot of cowboys out there that this is one thing that, the field suffers from, doesn't it, really? There are a lot of snake oilers.
Well, that comes down to another psychology paper published a long time ago that, what did it say? That competence is inversely proportional to confidence. So the smoother talking somebody is, probably the more incompetent they are, especially when it comes to selling you something. So anyway, so that was our discussion. I hope you enjoyed it. We'll be choosing another paper that fits in with our aging research rubric. And I hope to see you guys in a few weeks. Enjoy your 4th of July, those of you who have escaped UK tyranny. Thank you.
Oh, that's below the belt. And to everybody that didn't, I wish you a happy Traitors' Day for the 4th of July from Britain. And thank you very much to our colleague here as well, Professor Wovitt. Thank you very much. All good. And if you haven't joined his Discord community, please do. Because he gets $1 every time someone joins. It's not true, really. Otherwise, I wish it was. And that's it. Thanks, Oliver, and thanks everybody else for tuning in. And more on the way, yeah. And thanks to everybody who supported us, our patrons, our Lifespan heroes, and you know, being able to do these live streams is all thanks to their to your support. So yeah, if you haven't checked it out, check out lifespan.io and Leaf. We'll see you again.